Mr. Phillips, and today we're looking at the kidneys. So you need to understand there are two main functions of the kidneys. So the first function is osmoregulation. So this is maintaining an osmotic concentration of body fluids in a narrow limit. And this mainly is about controlling water content of the blood, which affects the water pressure. So what the kidneys do is they remove excess water and salt, and water and salts are involved in both blood pressure and because they affect the water potential of blood. So that will then affect how much volume of water is actually in it. They're involved in excretion because they involve the removal of toxic waste products from metabolism, being the main one being urea, which is broken down in a process called deamination in the liver, where it's created from the breakdown of excess amino acids. And this can also be from the breakdown of excess nucleic acids as well. So the kidneys are part of the renal system or the urinary system, and the blood flowing into the kidneys comes via the renal artery. The blood then leaves via the renal vein, and we'll talk about the blood vessels within the kidney nephron in the next few moments. The urine, which is urea, water and salts, leaves via the ureter, where it's stored in the bladder, before leaving the body via the urethra. So make sure you get ureter and the urethra the right way around. Here's a structure of a kidney which you should know. So on the outside there's a tough capsule around the kidney, and it's got a good blood supply. Then you have the cortex, which is this layer here, again with a good blood supply. The medulla, which is like the inner layer, and then you have the pelvis area here, which is where all the end of the nephrons connect to form and the urine leaves. So overall what happens in the kidney is the waste is filtered from the blood in ultrafiltration, which we'll look at in detail shortly. Any sugars, then glucose, is selectively reabsorbed. We should reabsorb 100% of it, and any dissolved ions that need to be reabsorbed are reabsorbed here. For the whole of the nephron, the water is also selectively reabsorbed into the blood. And then at the end you end up with urine, which contains water, urea, and excess salts. So now we're going to look at ultrafiltration and selective reabsorption, which you need to know the processes in detail. So we need to understand what a nephron is to understand this. So a nephron is a functional unit of the kidney where filtration and reabsorption occurs. It's made of a single layer of cells called epithelium cells. The nephron is like the alveoli for the lungs. It's the functional unit of the kidney. So there's millions of them within the kidneys. And the picture here, which you need to get used to, is it starts off in the Bowman's capsule where selective reabsorption occurs. You then have the proximal convoluted tubule where, the Bowman's capsule is where ultrafiltration occurs. You then have the proximal convoluted tubule where selective reabsorption occurs. Water is then absorbed in the select loop of Henle and the distal convoluted tubule before the collecting duct is a final control of water levels in the body using the hormone ADH. Here's another picture of it showing you the nephron. So we've got here, this bit's called the Bowman's capsule, then the proximal convoluted tubule, and then you go from the cortex of the kidney down into the medulla of the kidney for the loop of Henle, back into the cortex for the distal convoluted tubule, and then the collecting duct, which goes again from the cortex down to the medulla. Notice how many blood vessels are all wrapped around them, because this is essential for the reabsorption. If you look at the renal artery, you can also see that the afferent arteriole, which enters this area of capillaries called the glomerulus, is wider than the efferent arteriole leaving it. That means more blood can enter the glomerulus than can leave the glomerulus, and therefore it builds up a high pressure in this glomerulus area here, forcing materials into the kidney as filtrate. So how does ultrafiltration work? Well, ultrafiltration works at the Bowman's capsule. As I just said, blood comes in from the afferent arteriole, a wider lumen here, so more blood can enter this glomerulus area here and can leave via the efferent arteriole. That builds up a supply of pressure. This tangle of capillaries called the glomerulus is in the middle of this section here called the Bowman's capsule. And what happens is the blood materials are forced through these layers here, which allows them to be filtered. Now the filtering is controlled by the fact there are 3 distinct layers. So in the epithelium of the blood vessel there are holes here which you can see the materials can fit through. You then have something called the basement membrane, which is a layer of glycoproteins which allow filtration, and then these podocytes which allow, called finger-like projections, the blue bits here, allow gaps for the materials to go through. So if you were asked how is the nephron adapted to allow filtration, this is what you would say. The first layer is the endothelium of the capillary. Each layer of the endothelium cell has thousands of tiny pores called fenestrations with a diameter of about 100 nanometers. So you can see that layer here. We then pass through the basement membrane, which is composed of negatively charged glycoproteins and collagen fibers, and it's like a mesh, so it acts like a filter. And then the last layer in the podocytes, where you've got these finger-like projections which don't fit completely together, allowing materials to go through. Now, ultrafiltration, the holes are large enough to allow most molecules through, but the larger molecules can't fit through. So it filters due to size. And molecules with relative molecular mass of 65,000 or greater can't fit through. So the molecules that can't fit through are proteins, red and white blood cells. And here you can see some examples of different things that can get through, okay, and then there'll be things that can't get through as well. Now from here we get the glomerulus filtrate, and that is 125 centimeters cubed of liquid every minute. So it's a really fast, efficient mechanism, but obviously this is far too much fluid to be losing from your kidneys every minute, and therefore we now have to start reabsorbing materials we need back into the blood. So as said before, the pressure is high because of the difference between the arterioles. So the afferent arteriole is wider than the efferent arteriole leaving the glomerulus. So it's like a traffic jam effect, and it maintains a high pressure in the blood. So once we get into the proximal convoluted tubule, the fluid at the minute is virtually identical to the blood. You cannot afford to lose all the glucose, amino acids, and things that are in this fluid, so we have to reabsorb them in selective reabsorption. 80% of this occurs in the proximal convoluted tubule, and all glucose and amino acids should be reabsorbed here from the proximal convoluted tubule into the blood capillary using a mixture of active transport and facilitated diffusion. So how does it work? Well, if we look here and we start sort of here, it all works using an ATP in a sodium-potassium pump. So sodium in the epithelial cells here are pumped into the bloodstream. That means lowers the sodium concentration in the epithelial cell. So sodium is then drawn in from a high concentration in the proximal convoluted tubule to a low concentration in the epithelial cell. That comes through via a co-transporter protein. So it can only come across if it brings a glucose or an amino acid with it. That then lowers the water potential in this cell, which draws water from a higher water potential in the proximal convoluted tubule to a lower water potential in the epithelial cells. And then the buildup of glucose or amino acids in the epithelial cells means there's a high concentration there, so they move across into the bloodstream. On top of knowing the process, you also need to know that the endothelial cells have microvilli on them, so it increases the surface area. And obviously, due to sodium potassium pump, they're going to need mitochondria to generate the ATP. Actively transported using the sodium potassium pump into the blood using active transport. The sodium concentration then is lowered in the endothelium cells, and the pull of that concentration gradient from high levels in the distal proximal convoluted tubule into the cells comes via a co-transport protein, and it also brings in glucose and amino acids with it. This then builds up in the cell and it diffuses across as a result. So the level of reabsorption here is mainly passive osmosis via water, and only about 45 centimeters cubed then leaves the proximal convoluted from the 125 that enters. So we've lost a considerable amount of water in this first section. The urea will diffuse back into the blood as well, though, but this is not all of the urea, so we're still losing a lot of urea. Obviously, only about 50% could diffuse, as it is due to diffusion from a high to lower level. So the next stage is, well, how do we reabsorb more water? Because at the minute we're still losing far too much fluid. So the loop of Henle, again, is this part of the nephron, and it goes down from the cortex into the medulla and back up again into the cortex. About 45 centimeters cubed leave the loop into the loop of Henle every minute, and it is a U-shaped. It goes down to the medulla, and basically what it does is it creates very high salt concentrations in the tissue fluid of the medulla, and then this later allows water reabsorption as a result from the collecting duct as well. Now, there are two parts to the loop of Henle, the descending limb and the ascending limb. So the descending limb is the first bit and the ascending limb is the second bit, where the liquid goes back upwards into the cortex. But to understand it fully, we need to study the ascending limb first. First of all, you need to know the walls are impermeable to water, but the cells allow diffusion at first and then actively transport sodium and chlorine ions out of the fluid in the tube into the tissue fluid between the limbs. This causes a very high concentration of ions in the tissue fluid next to the descending limb, drawing water out. So what I'm saying here is sodium and chlorine are actively pumped out up here, diffused out down here, so you get very high concentrations of sodium and chlorine in the tissue fluid here. That lowers the water potential in this area, and then the water that's going down the descending limb moves out from a higher water potential to lower water potential via osmosis. So the descending limb is permeable to water as well as ions, and the water is drawn out via osmosis to the lower water potential. Also, the sodium ions here and chlorine ions will then diffuse back in here from a higher concentration to a lower concentration via diffusion. So the water comes out, making the water more and more concentrated as you go down. And you can see here the diffusion as you go down. So we get diffusion of sodium and chlorine out at the bottom, then it is actively transported out at the top of the ascending limb. Water diffuses out via osmosis from a high concentration to a low concentration caused by these ions that have been actively transported or diffused out. Now, because the water levels are decreasing, this gets the fluid getting more and more concentrated until a maximum here at the bottom of the loop. Because as you go up, even though you're not losing water, you then pump out sodium and chlorine. So the water potential actually starts to rise again as you go higher up in the loop. Now, also sodium and chlorine is actively pumped this way as well to this tissue fluid, which will aid the removal of water as you go along this area through the distal convoluted tubule and down the collecting duct as well. So the fluid concentration is very concentrated as it starts the ascending limb here, but the ions leave, as I said, so you get very high concentration to low concentration, and the tissue fluid concentration is highest at the bottom of the loop in the filtrate, which means it diffuses at first and then active transport as it becomes less concentrated as it goes up. Now, you could be asked to answer and explain this in a graph showing what happens at different points in the whole of the nephron. So in the proximal convoluted tubule, selective reabsorption occurs, so glucose levels drop, and the blue one here is sodium, so not a lot happens to sodium at this point, but urea concentration increases because there's water being removed, making it more concentrated. And that's why the urea keeps going up as we go all along here, because there's less and less water, meaning there's more and more concentrated urea. Sodium levels then in the loop of Henle increase as you go down the descending limb, because remember they're selectively reabsorbed back, they're diffused back in, but then it decreases as they're actively pumped out of the loop of Henle. And then as water is lost, and the concentration increases as a result because there's less water of both sodium and potassium tubule. And then that affects the water potential in the surrounding tubule. Now, you could be asked about how water is gained and lost from the body. So the three main ways in which water is gained and lost from the body is it gains food. So think about fruit's got lots of water in it. We drink it, and our metabolism, respiration, produces water as a waste product. We lose water in our urine, in our feces, in our skin, and in our breathing. So this was from a question, so this would have a graph on there as a result. So as we go along the loop, we're now going out of the loop of Henle into the proximal convoluted tubule and the collecting duct. The tissue concentration, again, ion pumping is used. So osmoregulation is used to regulate the water's concentration at this point. Water levels affect both volume of blood and pressure in our blood, so it's important. And we can control our water gain. We need to sort that out. Our water losses, these four main things. But precise control of water loss is controlled using hormones and the hormone ADH, or antidiuretic hormone. And the body uses this hormone to regulate solute potential of the blood to the water potential to precise levels as a result. And this diagram shows how it works. So first of all, we have our water levels are detected by osmoreceptors in the hypothalamus. And basically, due to osmosis, they either lose or gain water via osmosis. They tell the pituitary gland to release it's the posterior pituitary gland to release ADH, which goes through the bloodstream to the kidneys in the collecting duct, and they make the collecting duct insert aquaporins into the collecting duct, making it more concentrated and less volume of urine. So let's have a look at that in detail. So osmoreceptors in the hypothalamus detect water potentials. Remember, there's different nuclei in the hypothalamus. One of them is to detect osmoreception, and it detects low water potentials in the blood. And something called neurosecretory cells do this because water will leave them via osmosis into the blood supply. As they shrink, they detect that change and they pass ADH along a special axon to the posterior pituitary gland where the ADH is stored. It then gets secreted into the blood and then goes via the blood to the kidney, where it will attach to a protein receptor, transmembrane receptor, and activate a series of reactions which cause aquaporins to approach the cell membrane. So ADH is a hormone because it is a chemical. It is produced in the hypothalamus but stored in the pituitary gland, so it's released from the pituitary gland and it is then transported to the blood to the kidneys, which are its target organ. So inside the kidneys, what happens in the collecting duct is water is lost out of the collecting duct and goes back into the blood, making urine more concentrated. On a hot day or a day you drink very little, you become dehydrated. That activates the osmoreceptors in the hypothalamus. They then store ADH in the pituitary gland, which is secreted into the blood, and this will reach the kidneys where it alters the amount of water loss in our urine, increases the amount of water in our blood, so less in the urine, more reabsorption into the blood. And this is done by increasing aquaporins in the cell surface membrane. So on a hot day, that is what happens. When you have lots to drink, the osmoreceptors prevent the release of ADH. It's no longer secreted into the blood, and it doesn't cause water to be reabsorbed into the kidneys, into the blood from the kidneys. So a lack of ADH means the aquaporins do not move from the cell surface membrane and do not trigger the vesicles to move into the membrane. So on days like this, you can see no ADH, the tubules have less material going back into the blood, less water going back into the blood, and therefore less water is reabsorbed and the urine has higher volume and is very dilute. So this shows you how it works in a bit more detail. So number one, the ADH comes and attaches to a transmembrane receptor. That triggers a series of enzyme control reactions, so it will activate an enzyme which then causes the vesicles that contain the aquaporins to fuse with the cell surface membrane of the endothelial cells, and the aquaporins allow water to enter from the collecting duct through the cells and into the blood, meaning more water is reabsorbed into the blood, your urine becomes highly concentrated and less volume as a result. You lose less water. Again, just a different way here showing you the aquaporins are water crossing membranes. They transport proteins basically which allow water to cross them, and they basically form gated channels. So when the ADH arrives, it causes the aquaporins to enter the membrane, and water can then travel through and into the bloodstream. So you reabsorb more water with ADH into the blood, so your urine becomes highly concentrated and low volumes. And again, it's a different way. ADH causes aquaforming to bind to membranes again, allowing water to enter the epithelial cells, and then this moves via osmosis towards the blood down a concentration gradient. Alcohol can affect this though, because osmoreceptors basically get affected by alcohol. It inhibits the release of ADH, and therefore you can end up losing lots of water, which makes you more dehydrated instead of reabsorbing it into your blood, and that can be one thing that contributes to a hangover.